talk to your doctor about an abnormal area or pain you're experiencing. Now having to tell your loved ones that you might have breast cancer. In 2014, it's estimated that there would be 235,030 cases in the United States. Breast cancer currently is the second cause of death amongst women, and it's crucial that this health issue is taken with immediate action and focus on improved diagnostic techniques for early prevention. Mammograms serve as the first line of defense for the detection of breast cancer. The sensitivity rate is about 66%, and the specificity rate is of 92. However, mammograms are responsible for 30% of overdiagnosis and does not reduce the mortality rates. Not to mention, measuring dense breasts is very difficult through a mammogram. Women with dense breasts are 6 times more likely to develop breast cancer. Here you'll see a stage 3 breast cancer picture. You'll notice the lymph nodes associated, the cancer is present, and the fatty tissues. Over here you see the fatty tissue. These are difficult to examine. That is why we need different techniques to help women who have dense breasts. We can give a better diagnosis. Wouldn't it be amazing if we had the technology to attack cancer cells before they can mutate? There's a type of microscope called a Fourier transform infrared microspectroscopy. This tool is able to examine blood samples and many other proteins at once, which saves a lot of time and other tests that need to be performed. This technique is able to differentiate between cancer cells or benign cells and detect the special differences between the two samples. Using this specific technique, it shows the difference in the biochemistry of those with or without cancer and a coinciding change with the T and B cells. This test confers the ability to distinguish the biochemical difference from healthy individuals, including those with benign tumors. This method is affordable, simple, and minimally invasive test for the detection of breast cancer. Current research of analyzing DNA biomarkers has shown much success in identifying cancer cells and tissues. If we can identify biomarkers that are associated with breast cancer, mortality rates could drastically decline, as well as future cases. Examining the methylation levels of biomarkers gives an insight on at-risk breast tissue and early prevention. Biomarkers are released by tumors or from other tissues as a result of inflammation from tumors that are present. Understanding the molecular profiles of a normal at-risk breast tissue can play a huge role in identifying new targets for prevention. An improved model of examining breast cancer is called breast spectral lavage. This has recently been performed by analyzing the cytopathological changes in the quality and quantity of DNA and RNA. This method collects breast fluid from the milk ducts and is then centrifuged and processed. This test further explores the ability to examine breast epithelial cells for precancerous cells. Focuses on the methylation is done on 48 participants. These levels will appear high amongst those who have tumors present. 13 genes were detected and used for providing evidence for breast cancer prediction. Discovering the gene panel can detect early breast cancer, and that's the goal. This goal is to try to control the mutations. Biomarkers consistently have provided the ability to detect the behavior of cancer and prognosis into the future. Studying genes, metabolites, and proteins have shown much success in identifying abnormal cells. The ability to detect gene methylation in circulating tumor DNA has the potential for monitoring therapy, the progression of disease, and the recurrence of the cancer. Germline biomarkers are capable of predicting the risk of exposure to a specific disease. BRCA1 and BRCA2 specifically are both genes that play a role in reducing the development of growth for tumors. proprietary DNA. If a mutation occurs in either of these two genes, the risk of breast cancer increases. MicroRNA is another widely known biomarker. MicroRNA can detect the sensitivity of patients that have tested positive with the disease and specificity for those who do not have. This testing has shown you very high rates for both categories. ATR2 is a cancer biomarker. This is the biomarker that promotes a portion is cleaved by the protease and from the receptor and is detected in serum as a biomarker. There are high levels present of the ATR2. This indicates that there was poor prognosis of breast cancer previously. Here you'll see two breast cells. You have a normal breast cell and an abnormal breast cell. This one here has the ATR2 receptors. There's a few normal ones. You'll notice over here in the abnormal, there are many. So this would be due to poor prognosis. Metabolomics, which is a study of how the body metabolizes products, shows even a slight bit of phenotypic change through analyzing hundreds to 1,000 metabolites at a time, and also multiple cellular pathways. Metabolomics can also distinguish between cancer and normal cells. Breast cancer prognosis can be analyzed by comparing the metabolomics profiles and the survival rates. With the increasing rate of cancer, it's crucial to aim studies toward prevention and disease detection. This is the future of everyone's family. Somehow, we all will be impacted by the subject. Whether it's your mother, your daughter, or in the future, it's a scary situation. There are many techniques available to assist in the detection of breast cancer through the use of biomarkers. The studies of genomics, metabolomics, and proteomics show to be very effective in the sensitivity and the specificity. The field of science is constantly changing, and we need to adapt or be ahead of science. Scientists have revealed that biomarkers play a role in identification of specific genes associated with cancer. The early formation of tumors and breast cancer have been identified through analyzing the alterations in the gene methylation levels along with the biomarkers. Further studies of biomarkers and their use could lower the mortality rates, prevent future cases, and reduce overdiagnosis of many cancers. Thank you, and I'll take any questions for those who have them.
Yes, what is stopping this practice from being used today?
Which one specifically?
The use of biomarkers.
I would say the reason why it's not being used as much right now is that this study is still very new. It was done between 2013 and 15. So I believe with more studies showing that it is effective in identifying cancer cells, that it will be incorporated and accepted, for an author on another approach.
And has there been any positive impact with this new practice as far as using biomarkers for early detection?
I believe so. It's the testing showing that the sensitivity and the specificity rates are very high. I believe it says 92%. So if using the biomarkers can increase the sensitivity, which is showing who has the disease, that we'll want to use it later on because it's going to show that we're able to detect those with the cancer, and that's what we want to do.